Biotechnology and the advent of traits has really revolutionized the seed industry, and at Stine it's been no different. We've had to respond to that, and we have with our own biotechnology facility. I've got Dr. Bruce Held here, who's one of the co-directors of Stine Biotechnology. Bruce, can you tell me a little bit about what goes on at this facility?
Yes, we've been here, David, for over 15 years, and we specialize in this process called transformation, which has created those crops we're all familiar with, the herbicide-tolerant crops and the insect-resistant crops that contain Bt genes.
Okay, so how do you get those traits into the plants?
That's a good question. You have to get the genes through the plant cell wall, which is a huge barrier to this process, and that's the first step in the process.
Okay, so what kind of methods do you use to do that?
Well, you can use a biological method, agrobacterium, or you can use physical methods, and in particular, we use the Aerosol Beam Injector.
Okay, which is this device here.
Exactly.
Can you tell me a little bit about how this machine works?
Sure. This is our proprietary device, and it works by first having a DNA solution metered into the top of the device, and here we create an aerosol, and then in this bottom chamber we'll put the plant tissue, which will be transformed, and we'll pull a vacuum in this bottom chamber. This vacuum will pull the droplets down through this small orifice, and when they enter that orifice, they're accelerated to supersonic speeds.
Wow.
And that's what gets the DNA into the cell.
So you just shoot a stream of DNA right into the plant tissue.
Exactly.
Now, how long does that take?
Well, this process here just takes a minute or two, and it's really short compared to the entire process of actually once getting the DNA into the cell and then selecting a cell with the DNA in it and regenerating a plant from that single cell. And that whole process can take 6 months to over a year.
Really? Okay, so it's not an overnight process.
No, it's not.
What kind of plant tissue do you use then to insert the trait into?
Well, we use a soybean embryogenic callus and corn immature embryos.
Oh, okay, okay. So what's a device like this mean for Stine Seed Company? What are the benefits of having this technology, which is our own, transformation technology?
I think the main benefit, David, is it gives us freedom to operate. And it gives us unique capabilities to work with other traits that maybe nobody else, are working with.
So you can work with genes that you've discovered in your own lab, or you can also work with other companies who have a trait of interest that they want to get into genetics.
Exactly.
Fantastic. So Bruce, what's the payoff for our grower customers?
David, it's new traits in Stine genetics.
Great. Thanks, Bruce.
Yeah, you're welcome.
So Stine researchers are working to develop the traits that growers want, packaged in superior genetics from Stine to provide a choice for maximum yield and profitability.